Next, it's my pleasure to introduce Tatyanna Sharpee from the Salk Institute.
Okay, thank you very much for the invitation. I'm looking forward to. Sorry. So as you can see, I modified the slide, the title of my talk to engage discussion. So I'm going to present to you hyperbolic hypothesis by, I think, let me be a little bit assertive and say all data in the natural world and in neural systems have hyperbolic geometry in them. And actually how we can use to quantify how much depth does the surface data provide. So let me tell you why I'm fascinated by hyperbolic geometry. Many reasons, three actually. So this is the first reason. The first reason is basically mathematical reason that every time you have a hierarchical system, it implies hyperbolic geometry. And I will define, I will give a background about hyperbolic geometry, but here is one. So suppose we have a phylogenetic tree, and you would like to, I don't have any. I think I will do the finger extension right here. So you would like to evaluate distances between two nodes on the network. Of course, we are not going to go this way. We are going to go towards the last common ancestors and back out again. So it means that somehow this is our notion about how we evaluate distances in trees. But in parallel, there was the idea of Poincaré, who created, he thought about hyperbolic geometry in the context of general relativity. But you can also create a plane where the size of the square divides in two. So supposedly they are the same size, but approaching the line of y equals 0, you will have infinitely small squares. So the metric, the distance, kind of becomes harder to move along the dimension of y equals 0. Yes?
So do you mean by this like negative Gaussian curvature?
Yes. So one can also evaluate that if I think about points A and B and I want to minimize the number of squares I'm crossing, then I will go back out and back in, and that's one can mathematically, analytically compute the trajectory, the geodesic, and it will, in this case, will be a circular trajectory that approaches infinity to at orthogonal degrees. So that was a statement, but a little bit of background about hyperbolic geometry and how it differs with Euclidean geometry. Maybe maybe people know already, but so we know Euclidean geometry that we studied in school. So of course, Descartes' coordinates. Do you know how he discovered his coordinates? So, so he was a neuroscientist at heart. You might not know this. So he was sick, and he was watching a fly on the ceiling. And this is when he had the idea that the distance between the fly and the corner can be estimated as the distance to one side and another side squared. So that's kind of a funny story. But it turns out that if you were to measure his perception and our perception of distances, it wouldn't be actually Euclidean. It would be hyperbolic. So artists, of course, think about this more. And you might know there is a series of paintings by Escher using hyperbolic geometry. And what the design is meant to capture that we have a center that we devote our full attention, and then everything else kind of gets progressively degraded. So he generates this with a hyperbolic. This is another representation, also Poincaré representation, known as Poincaré circle, where you take infinite plane and you compress it with a logistic nonlinearity into a finite circle of radius 1. So this size over here is unattainable, but so that's one, how artists try to summarize our perception. And it turns out that for us, there are several pieces of evidence that our visual perception is hyperbolic, but one is that children try to grab onto the moon. So psychologically, the moon is actually much closer than in physical distances. That's one example. Then there are many, but there is another example that if you want to have straight lines in a building and you're not moving your eyes in a room, you actually have to curve the walls. Vice versa, if you measure the percept of straight lines, then you will be curved. And it turns out everything new is well forgotten old. So it turns out Greeks knew that. And so when they build their temples, they will shave the columns and so on in a way that to achieve flat, you have to curve such that when people look at it, they will perceive. So this is some circumstantial evidence, but I will present more quantitative evidence. But right now, I wanted to talk about why some of the advantages of why everything is hyperbolic. So since we have machine learning people in the audience, I was going to quote these results on connectivity in how to navigate in networks. And there is a fascinating set of papers by Dmitry Kryukov and collaborators. So where they talk about in order to how to communicate in a network when you don't know the status of the nodes, full connectivity structure of the network. And it turns out that if the network has hidden hyperbolic structure, then you can navigate using local cues, meaning I know my coordinates, I know coordinates of my target node, I know coordinates of nodes around me, and I send along the shortest path in that latent space. So that's their results. And what I will also talk about at the end of my talk is actually that hyperbolic geometry is important for motor control. So we talked about how sensory and systems and motor systems are coordinated. So there will be a theoretical result about this. In my own group, we have studied hyperbolic geometry in many different contexts. So in plants, I will describe some of these results in more detail, but we have found them in natural olfactory spaces, order statistics, so like metabolic signals, in gene expression, in neural representations, and this is, I will also expand today about this. And then there we see that neural representations actually grow proportional to how much entropy you inject into the system. In COVID evolution, viral evolution, and also in aging. So I'm not going to go through all of these, but I will give you some example from odorants, from neural representation, one slide on COVID evolution, and then talk about relationship with motor control. So in olfaction, we had a talk about language. Well, olfaction is also a communication system. So I smell a sandwich, and I want to figure out whether it's OK to eat it. So, through experience, if it smells like this, better not eat it. Maybe some poisonous bacteria there. So olfaction is interesting because it is a communication system, and it has puzzled people for a long time. This is a figure from a review by Salk, who studied olfaction in the industry. And he came to the conclusion in this review that olfaction basically cannot really be solved in the near future. So he would say, here's an example of these molecules where triplets of molecules. So one or two of them will smell similarly but have different structure. and 2 of them will have similar structure but smell differently. And this is almond smelling hydrogen cyanide. So apparently if somebody gets poisoned, then they smell like almonds. And actually that's why almonds have in them and have small amounts of cyanide in them. So the data science or computer science approach to this, or physicist approach to this, you have these genetic varieties of strawberries of all kinds, and they are interested in making a good strawberry for us. So they measure different genetic varieties of strawberry and what molecules they emitted. So small data set, but about 60 molecules. So you can evaluate a statistical distance between molecules, meaning if something is correlated, then they will be more closely related to each other. And in this way, you define distance. And using these distances, you can ask what surfaces does this distance matrix imply. So we have a matrix of distances of correlations in the data.
And it's a Euclidean distance.
Yes, correlation. Okay. So you can do. So it's an interesting question. It can be said that there is some kind of an inconsistency there, which we thought about it, and I have an answer. I can give it now. So imagine that, especially, for example, in gene expression, or here, you imagine that the data lives in a low-dimensional hyperbolic space. But I don't know the space. So I try to take many different snapshots of the data. So I'm taking lots of measurements. So suppose data is 5-dimensional hyperbolic space, but I took 20,000 measurements. Then I evaluate distances in the Euclidean space. So it's like I'm embedding this 5-dimensional hyperbolic surface in a high-dimensional, 20-dimensional Euclidean space. Turns out that if I do this, and the dimension of my embedding is higher than, or much higher than the real low-dimensional curve dimension, I will have an accurate embedding. So after that, I can take these Euclidean distances and attempt to put them in the low-dimensional hyperbolic space. And it will be OK. But if I make small number of measurements, not 20,000, but 5, in the process of measurement, I flattened my data, because I put it in a flat Euclidean space, and there is no way after that to recover the curved structure.
So the question is, what do you do first? Do you first measure the Euclidean distance and then project it to a hyperbolic, or do you look for the hyperbolic projection that will minimize the distance?
Well, the first we take a large number of Euclidean measurements. So you embed into a large space with, well, I think the process of measurement is like an embedding. I evaluate distances in Euclidean way in many dimensions. It still preserves the curved inner world. And after that, you can try to do different embeddings, optimize the curvature, optimize dimensionality, and find the right one. And then it turns out that if I, for example, under or overestimate curvature, depending on the method that we use, for example, non-metric multidimensional scaling, then you will have some signatures that the curvature is mismatched between the embedding and the kind of the real hidden structure. So I think this is a non-trivial question that I think can be addressed by, in other words, so for example, in gene expression, if I have 5 very important genes, and I measure these 5 genes, and with respect to these genes, the dimension is surface is hyperbolic, I will be OK. But if I measure 5 random genes, I will lose the hyperbolic structure. So as long as we measure lots of things, it should be OK. So then the goal is to match the distance matrix of the data with the distance matrix of the geometry we can try different curvature, both negative and positive or zero, and different dimensions. And in playing around with these things, we have a suite of measurements, either topological or multidimensional scaling with hyperbolic metric, or t-SNE with hyperbolic metric, or in the extensions with larger data sets, because some of these methods are limited into how many points they can embed, and the newer methods can take up to 40,000 points.
So it's constant curvature, or is it just for this, just for the drawings?
This is just for the, so the algorithm can adjust curvature, but we only consider spaces with constant curvature. In reality, it's not, I don't think it's constant, but it's an approximation. So another model of hyperbolic geometry that is also tree is like a broccoli. And imagine that you give more information to one part or more nutrients to one part, so it's going to branch more there. So in reality, I think the curvature is not constant, but as a first approximation, we treat a constant curvature.
So wouldn't it be better to measure the Euclidean distances only locally before you project and not globally? Because then it would be, I mean, I think the global measurements are the one that distort, right?
Well, so
hyperbolic is
you're right. So in fact, when we but using, it turns out that so when we look at geometry, for example, on gene expression, it turns out that locally it's Euclidean, globally it's hyperbolic, because locally you have noise that kind of washes out the structure. So I think that is a correct perspective, and in fact, in this hyperbolic t-SNE, we it kind of assumes locally Euclidean geometry and global hyperbolic. But when we evaluate distances, somehow I was always in favor of taking lots of measurements and not. I think the way to maybe to rephrase your question is to say, suppose I pre-cluster my samples and then evaluate distances locally within the cluster, but then maybe something we can be think about that. But for now, we did Euclidean distances along many dimensions. So this is an example of strawberry. So we are comparing three surfaces. And in this particular example, we used topological method. And according to these topological signatures, number of cycles, one can see that the hyperbolic geometry fits the data in 3 dimensions, 3 hyperbolic dimensions. But you can rule out either spherical or flat geometry. And speaking of surface data, when you do the embedding, it turns out that for these orderants, all the points are on the surface. And the reason, the way we think about it, is because these are olfactory volatile molecules that flew away from the plant, in this case, strawberries. So, but you're interested what's inside, what's not volatile. So taking these surface measurements and evaluating how correlated they are, or what is the distance between them, can give you insight about the structure of the hidden tree that is not measurable. And so the more measurement you take, the deeper into the tree I think we can infer. And incidentally, when we analyze different varieties of, say, fruits, strawberry, blueberry, tomatoes, mouse urine, they turn out to have the same curvature, which one can interpret as having the same complexity of the underlying tree. So roughly, approximately saying that the metabolic networks that are underneath have similar capacity. So that's an example from olfaction. I will try to briefly summarize the neural responses, because neurons are changing, are plastic, and there we find that the neural representations actually increase, and they increase proportionally to the information that was injected into the system. So in this case, it's data from the hippocampus, the area of the brain that represents space. And you have a set of neurons. So one neuron will be responsible firing at one part of the space. There will be another neuron in a different part, and they all have different sizes. And turns out that using this collection of sizes, you can construct a hierarchical network. So it's a mathematical construction where you take the size of the circle as the z-coordinate, and so small detailed response regions have low z, and large circles have large z. So I'm just meant to show that this is an equivalent representation either as overlapping set of response regions or as a hierarchical network. Any questions? Okay. So it turns out that apparently Darwin knew about this, and he used it to construct a set of his tree of life. So prior to Darwin, people were trying to classify species into these overlapping circles, depending on how petals, how many petals the flower has, and all the kind of phenomenological description. And so that's with mathematics, same representation, but a different idea. And so in the case of the hippocampal data, you can, in this case, there are several data sets we worked on. One data set is a linear track, and you can measure the response regions. So one neuron, some neurons have small response regions, some have meter-long response regions. There is an exponential distribution of these response regions, which is consistent with hierarchical representation. And then I will just maybe skip a little bit the data in the interest of time, and just how many minutes? 8. Okay. So this is I will just show you. This is the data with topological methods showing that the data is consistent with hyperbolic geometry, and you can rule out Euclidean geometry, and there are multiple data sets, linear track or a square track. And the interesting part here, because these are neurons, so one can analyze each session separately, and that what you find is that the curvature or the size of the representation is slightly smaller for first exposure to the environment and larger for when the animal is brought repeatedly to the same environment. So, and it fits with the logarithmic expansion. So you take, so the data set consists, the animal is running around environment. So it's, in this case, it's about a meter long white box. The animal is running around, and there are electrodes, and there is data from between 50 and 100 neurons at the same time. You measure distances as correlations between these activity, which neuron correlated with what, and you created this distance matrix, and then we try to put this distance matrix on different hyperbolic surfaces. So in this case, a three-dimensional hyperbolic surface with different curvature, and optimize the curvature until the distance matrix is reproduced. Okay? And then it turns out that if this is done for early sessions versus late session, the curvature is larger for the late sessions than for the early sessions. And the dependence is logarithmic with time. This is a fit using dashed line. And one can understand why the logarithm, because logarithm actually represents, or the entropy, the maximal amount of entropy that you can acquire in chunks of like discrete episodes, say t0, is given by log 1 over t0 plus corrections. So in the limit of large exploration time, you can see that the expansion of a tree, which is what we measure with curvature, is proportional to the entropy. And actually, it goes all the way back to the original definition of information in the Shannon's paper. So there are 3 postulates that he used to derive logarithm for the entropy. One, that if you add more measurements, the entropy will increase. The other one is that independent measurements add. And then the third one is that if you have a tree-like structure, then the entropy at the end should be additive with the entropy of the subtree. So I think it's not a coincidence that the entropy and the information and the hierarchical trees are all related to each other. So then one more fact about neural responses. One can ask, what is there an, yes?
I'm just thinking about this statement. What is the new information? You have a fixed box, and very quickly you explore it fully.
Yes.
So just when you keep running around in the box, it doesn't give you any new information.
Yes, it does. So I will skip to the summary slide of this section and then go back to the previous one. So what happens in the hippocampus or in general, even though it is one box, you form more detailed representations. So there is this movie, Monte Cristo, where he asked him about these stones and have you named them. So with time, there are 2 ways of thinking about it. One is mechanistically. So it is known that with time the receptive fields get smaller, and you can also have new response regions. So in this way you can see that representations get more detailed and there is a bigger depth. But there is another explanation that what is the optimal size of, what is the size of the place field? Can I write? Yes. So suppose my overall observation time is t. This is how much time I spent in here. And there is a minimal time t0 that I should be within a certain region of space in order to justify the existence of that place field. Okay? So then t, roughly, if they're all the same, t over t0 is the number of these cells. Now in an exponential hyperbolic space, most of the data, most of the weight is on the boundary. So you have that e to the r, which is the size of the space, goes as n, which goes as t over t naught. And from this you get that r goes as logarithm over t over t naught. Okay, so e to the r goes as n, goes as t over t naught, or r goes as log of n, goes as log of t over t naught. So even though, we could say there isn't much there to learn, but if I come to this room time after time again, I will start noticing a smaller detail. Which chair has this defect, or I like sitting here. So they all, does that, that's, and I would say, that, in the talk about, what does it mean? You have this complicated scene like walking tours, how to quantify. Well, it depends on how many sensors. You can say, I have this many sensors, so I'm going to split this scene into this many objects. And the more I look at it, the more objects I can identify. No? That's 1 minute. Really 1 minute? Okay. Well, then I will skip one part. I will just say that the number of neurons turns out to be optimized. The curvature and the number of neurons are related. So if I have too many neurons, if I have not enough neurons, then I will not form a detailed representation. Then we've seen the same expansion, actually completely different domain in viral evolution. So a virus is exploring its space. And when you fit the data, it's hyperbolic and the curvature increases. then there is a vaccine applied at 40 weeks. This is in this data. And you see how the curvature drops. And the same logarithmic expansion also applies in the viral domain. And in 1 minute, I will just intrigue you by saying how this relates to the motor system. And I will share you a fact that is known, but to me it is a remarkable discovery. How do we define distances between two Gaussians? Okay? A standard way is to use Fisher information. Okay? So what does it mean, the Fisher information? It means that the distance between the means depends on their variance. In other words, when the two broader Gaussians, so that's the metric, and actually that's the Poincaré metric. So if you think about, where is my slide? So if Poincaré plane is a plane where mean of the Gaussian is on the x-axis and the width of the Gaussian on the y-axis. So a broad Gaussians live here, and a narrow Gaussians live here. So the distance between 2 points here is smaller because it's measured in units of the variance of the Gaussians. And so the Fisher information here for the family of Gaussians actually is the Poincaré plane. And it turns out that the optimal way, if I want to move hand from one position to another position, is by following the geodesic, because that's what minimizes the dissipation. And this is other people's work, but they figured out that when you move the Gaussian, the optimal way is not to drag this Gaussian like this, but the optimal way is to, you start with, say, this distribution, you have to broaden during transition, and then you have to collect the data. So the optimal way of broadening is described by this geodesic in the hyperbolic space. And it turns out there are some quirks about it, that if you have to move too far, you can't, then your optimal solution will spread it out so much that you won't be able to physically put it back together. In this case, the optimal solution is to make a small jump, recollect, small jump, recollect. And we are looking for evidence in, say, biological systems when the systems differentiate in cells. They go through all kinds of checkpoints to kind of regroup the variance. So, and actually, since we were talking about hands, about how important they are, so it turns out, I'm going to mention to you that there's this observations that if you think that you perceive hands as they are, that's not true. So there are all kinds of distortions. The hands are the actual hand is like this, and there are distorted shorter here and longer there, and we are working to figure out whether this can be explained by hyperbolic geometry. Also, there are distortions in the face. So with this, I will summarize by saying that efficient representation of signals benefit from hierarchical or hyperbolic geometry, that representational capacity increases with curvature. By curvature, we mean the size of the space relative to the unit curvature, and that increases logarithmically with time and is proportional to the information that is injected into the circuit. And that hyperbolic geodesics are actually optimal control trajectories. So and that's various people that on our hyperbolic journey. So Yuan Cheng-Zhou did olfactory work. Hippocampus was done by Hongqiu Zhang. Anup Praturu developed methods that scale, and Minqian Yao is continuing this, graduated recently. Thank you so much.
Maybe take one more question and then go to coffee. So pick your...
Okay, well, you
pick. So how do you practically move? I mean, let's say I have lots of data and I sample the distances, I measure the distances in the Euclidean space. How do you practically move it into the optimal hyperbolic? I mean, how do you determine the parameters? What is the algorithm for moving from one space to the other?
Okay, many choices. We use multidimensional scaling, non-metric multidimensional scaling. You can do
No, but do you predetermine the parameters of the hyperbole, or do you?
no,
or are these
you
searched?
we optimize. So the dimension is more complicated. You try different dimensions, and then curvature. You can do a Bayesian estimation with a prior. You can so you just maximize log likelihood of the data given these distances, and we have choices. We can do topological methods. They have advantages and disadvantages. For example, the distance might not be the real distance. We might not know whether Euclidean distance is the real distance. So the advantage of using these topological methods is that they allow for like non-monotonic, from monotonic transformation of the distance and still be okay. But they do not work beyond 100 points. So then you do multidimensional scaling, and there we try to do one method uses the actual distances, and the other one is a non-metric version that tries to say we don't really know, if it is distance or e to the distance should be, should work similarly.
Is it not true that a lot of the measurements you're taking are from physical world?
Yes.
So you would have a lot of epistemic uncertainty and you would have allosteric uncertainty. Right? So noise in different forms, right? So for example, if you're looking at, say, gene expression
Yes.
information, that is just a sort of, it's a proxy for protein expression.
Yes.
So you have epistemic uncertainty.
Yes.
You would have noise in a lot of other measurements. And
Yes.
this would be a very important criterion in determining what is the nature of the underlying manifold.
Okay, I can tell you the answer to. We studied this question. So it turns out that if you denoise the data in various ways, then, for example, the estimates of the curvature increase by 10%. So the more that's one statement. Second statement, as I mentioned briefly, local. You can think of what does it mean hyperbolic. If I have a lot of correlations in the data, these are like branches of the hyperbola. And the noise will smooth them out, so they're like swimming in the sea of noise. So the more noise you have, the estimates of the curvature will be less. And then if you denoise the data, the curvature increases. And if you look locally in the space, then it will be more Euclidean data. So in the case of gene expression, so, for example, this Bayesian method, it will go down to zero curvature, or essentially zero curvature. And so locally, if you look at cells within one organ or like one type of cell, then it will be Euclidean representation, small curvature. If I start incorporating multiple different cell types, the curvature will be bigger. If we look at, we can estimate the curvature, for example, for cells that are brain cells. The curvature there is bigger than the curvature of cells in the lung or in the kidney. And if it is more these stem cells, then the curvature is even less. So it's a parameter that is tunable. And you can see how hyperbolic is the data. For example, if the curvature is less than 1, then I would say it's almost Euclidean.
This is the point
Okay,
of...
we're going to
Yeah, we can take this
stop
offline.
it
Yeah.
right here. We're going to take
Sure.
a break. Thank you so much.
Thank you so much.
We're going to take a coffee break. We'll be back at 11 with Dr. Malik.
Okay, so I would like to answer your questions better.
11:00.
11:00.